Live bacterial spores displaying antigen or enzymes are functionally active nanoparticles that can be used as mucosal vaccines or biocatalysts. In principle, any protein can be efficiently adsorbed to spores.
This technique is simple and does not require any genetic manipulation. Therefore, no release of recombinant organisms into the environment is involved.
In addition to mucosal vaccinology for humans and animals, spores displaying enzymes can be developed as reusable biocatalyzers.
Begin by labeling 2 × 10⁹ B subtilis wild-type purified spores with 2, 5, or 10 microgram concentrations of model RFP in 200 microliters of binding buffer supplemented with 50 millimolar sodium citrate for 1 hour at 25 degrees Celsius on a rocking shaker. At the end of the incubation, pellet the binding mixtures by centrifugation and transfer the supernatants into new conical tubes for subsequent adsorption efficiency analysis. Then wash the spore pellets 2 times with 200 microliters of binding buffer per wash, resuspending the pellets in 100 microliters of fresh binding buffer after the second wash. For surface protein extraction, add 50 microliters of each adsorbed spore resuspension to 50 microliters of 2x sodium dodecyl sulfate dithiotheratol to solubilize the surface spore proteins. After 45 minutes at 65 degrees Celsius, collect the mixtures by centrifugation and run 10 microliters of each volume of extracted protein supernatant on a Western blot using a